Hello, everyone, and welcome to today's live broadcast, Simple and Effective Tools for Antibody-drug Conjugate Screening and Characterization, presented by Chris Langsdorf, a staff scientist for Thermo Fisher Scientific. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, visit thermofisher.com. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Chris Langsdorf. I will now turn the presentation over to him.
Thank you, Judy, for that kind introduction and for hosting us today. As Judy mentioned, my name is Chris Langsdorf, and I'm a staff scientist at Thermo Fisher Scientific in our protein and cell analysis group. And today we'll talk about simple and effective tools for antibody-drug conjugate screening and characterization. Now, in our group within Thermo Fisher Scientific, we developed fluorescent reagents, all of the probes and assays we use to characterize our cells using fluorescence microscopy and flow cytometry. And at a basic level, much of the work we do and much of the work in industry and in academia right now is based on this interaction of an antibody and a cell and how they interact in various ways, whether that's by binding or by the antibody after binding having some modulatory effect on the cells. Now, an enormous amount of energy and resources is focused on this simple interaction. Today we'll look at how we can go one step beyond an antibody binding to a cell and look at how we can weaponize that antibody to create an antibody-drug conjugate. Now, by conjugating a toxic drug to an antibody, it gives us the best of both worlds. The advantages of an antibody being specificity, a long half-life, and the in vivo biodistribution of an antibody, while at the same time giving us the potent effects of a toxic drug delivered specifically to our target cells. Now, for this reason, there's intense interest in developing antibody-drug conjugates to treat a variety of different conditions. Now, in the process of developing antibody-drug conjugates, one of the critical answers during that development process is, does my antibody internalize into our target cells? Now, of course, early in development of an antibody-drug conjugate, we need to know if our antibody can effectively bind with high affinity to an antigen on the surface of the cell. But also, critical to success of an antibody-drug conjugate is whether, after binding, that antibody can efficiently internalize and be specifically taken up by the target cell such that the conjugated toxin, shown here in red, can be released and specifically kill our target cell. And so historically, this has been a very challenging event to measure with various approaches attempted, all with their own drawbacks. And so today we'll focus on 3 different methods to evaluate antibody internalization. 2 that are covalent attachment, one that is random conjugation and one site-specific, and then a new method that is intended for fast, scalable screening of this process of antibody internalization. So I'll show you examples of how we apply each of these technologies to evaluate the performance of our antibodies and antibody-drug conjugates. So First, let me show you the cell models we used in our lab to interrogate these processes. First, we have a blood cancer model with two very well characterized therapeutic antibodies, rituximab and obinutuzumab, both of which bind to the CD20 antigen on the surface of cells, such as these Ramos leukemia cells. Our second model, which is an adherent cell model, uses trastuzumab, or Herceptin, which binds to the HER2 receptor, as is present, overexpressed on the surface of these SKBR3 breast cancer cells. So both a suspension cell model and an adherent cell model. The suspension cells we analyze typically using flow cytometry on our Attune NXT flow cytometer. Now this instrument has 4 lasers, 14 colors, and with an auto sampler, we can run 96 different samples very quickly. Critical for the screening aspect of the data we'll show you today. Our adherent cell models we analyze using an imaging system, the EVOS FL Auto 2 imaging system, for qualitative data. For quantitative data from images, we use our CellInsight CX7 high content analysis platform, which extracts quantitative information from the images of our cells. And so to actually monitor internalization of our antibodies, we take advantage of 2 different fluorescent dyes called phrodo dyes, both of which, a red and a green, both of which are dark at neutral pH, and as they encounter an acidic environment, become dramatically brighter, as you see in the figure on the right. And so in the panel on the left, when we label an IgG antibody with either of these dyes, it will be non-fluorescent outside the cell. Only after that antibody binds to its antigen and is specifically taken up through the endosome into the lysosome, where it experiences a pH drop and becomes very brightly fluorescent. And so we can quantify that to determine the extent of antibody internalization. Now you'll notice the acronyms IFL Frodo Red and IFL Frodo Green. This is to distinguish newly developed versions of the Frodo dyes, which are optimized for antibody labeling. The name Frodo has been around for many years, but the previous version of these dyes was not ideal for antibody labeling. And you can see here as overlaid the classic versions of both Frodo Red and Frodo Green were not useful for monitoring antibody internalization. So for this type of assay, be sure you are using the IFL Frodo Red or IFL Frodo Green. The reason for this has to do with solubility and performance of the antibody during the conjugation process. If we look at, back up a step and look at the actual conjugation of the trastuzumab we used in this experiment, we can look at the spin filters used to clean up this conjugate. And you can see with the classic Frodo Green and Frodo Red, we have a lot of color in the top of this size exclusion column, showing us that all of our antibody and dye precipitated and did not flow through the column, whereas both the IFL Frodo Green and IFL Frodo Red, we have our free dye collected on top of the column, and our purified conjugate flows through and is collected in the bottom of this column. And so with the new IFL Frodo Red and IFL Frodo Green, it's very easy to conjugate antibodies or any other globular protein to monitor its internalization. And so what we see when we take, for example, this Herceptin labeled with IFL Frodo Red and put it onto breast cancer cells overnight for 16 hours, what we see in a live cell image is a red signal present within the lysosome of every cell, reporting very robust internalization of this antibody into the target cell. So very, very clear fluorogenic indication that this antibody has been internalized, which we can see qualitatively in this image. And so using flow cytometry, we can provide a quantitative analysis of the internalization of these antibodies into our target cells. So on the left, using IFL-Frodo red conjugates of 3 different antibodies, Rituxan, which is a type 1 IgG. We expect that to internalize very rapidly. Gazyva, being an IgG2, internalizes to a lesser extent, while Herceptin, which does not recognize an antigen present on these cells, we see a fluorescent signal similar to the background of unstained cells. And with the IFL-Frodo green, we see a similar performance, except you can see that the background is a little bit higher with the green. So if sensitivity is the most important aspect of your experiment, we recommend the IFL-Frodo green as being somewhat brighter, while the green is available as an option if green is an ideal color for your detection system. And so in this experiment, we analyzed our CD20 positive Ramos cells on the Attune flow cytometer. We can also derive this sort of quantitative information from images. And so now we can switch to our Cell Insight CX5 high content analysis system and look at HER2 positive and HER2 negative breast cancer cells, which were treated with either IFL-Frodo red Herceptin or Gazyva for 24 hours. And what we see as we increase the dose of our antibodies from left to right, we have a very clear signal showing specific internalization of the Herceptin into the HER2 positive cells, while our various negative controls give us the expected lack of fluorescent signal. And so this is one way to show specific internalization with various controls as extracted from an image. Another way to show specificity is with a competition assay. And so in this experiment, we pre-treated our cells, our SKBR3 cells, with an increasing dose of Herceptin as a pretreatment. So as we move from left to right, we increase the pre-treating dose of unlabeled Herceptin. And then we chase with 5 nanomolar Herceptin, which has been conjugated with IFL-Frodo red for 24 hours. And so what we're asking is, can we use the unlabeled antibody to compete off the labeled Herceptin and block its internalization? And as you see, the answer is yes, we can very effectively block the internalization of this antibody by saturating the receptor. And so valuable to show that any internalization we see here is specific to the biology and not from nonspecific endocytosis or other sources of background. We can also look at the rate of internalization using the same system. So in this case, we're again using Herceptin with our SKBR3 HER2 positive cells or another HER2 negative cell line. And as we'd expect, the intensity of spots in a ring around the nucleus, as quantified by the Cell Insight CX5 platform, increase over time until we hit a plateau at around 20 hours, which is consistent across our other experiments and shows, again, the specificity of the biology we're looking at. And so one other thing to be aware of is these antibodies I've shown you so far have been cleaned up. I purified these antibody conjugates on a size exclusion column to remove the free dye. We wondered, is it necessary to remove the free dye? Or if dye remained in the conjugate, what effect would that have? And so we treated our Ramos B cells with increasing doses of Rituximab labeled with IFL-Frodo green, and we see the signal increase as indicated by the green line, as we'd expect as we increase the concentration of that antibody. If we treat these same cells with the IFL pHrodo green dye alone, we actually don't see a signal until much more free dye is added. And so the conclusion here is that free dye that's remaining in your antibody conjugate will not significantly affect the internalization signal as reported in your experiment. So this adds some flexibility or convenience where you may not need to clean up your conjugate as rigorously if you're concerned about compromising yield of your conjugate. Okay. And so that's a summary of our IFL pHrodo amine-reactive dyes for antibody internalization. But other conjugation approaches are possible. So I'll show you two more. First, I should mention that antibody-drug conjugates are generally made one of two ways. Either their lysines are labeled via a primary amine conjugation, or disulfide bridges are reduced to conjugate to thiols present in cysteine residues. Now, with lysine conjugation, this is essentially painting dye or a drug onto an antibody, whereas you see in this graph, the number of drugs per antibody varies widely along a normal distribution. While with cysteine conjugation, when we reduce disulfide bonds, we're essentially freeing either 2, 4, 6, or 8 conjugation sites, and we see the consequent number of drugs per antibody. With both of these approaches, we don't have, as you can see, we don't have control over the degree of labeling. And also, we may be concerned with lysine conjugation that a dye or a drug present in the antigen-binding site may affect the affinity of our antibody. While with cysteine conjugation, we can expect over-reduction of disulfides to compromise the integrity of our antibody. In either case, this makes it more difficult to pursue regulatory approval for one of these compounds as a therapeutic. Ideally, we would have a known number of conjugates per antibody and in a known location, so we can very tightly control the conjugation of our antibody and more easily gain approval for this therapeutic. Now, various approaches are used in the field to perform site-specific conjugation to attach drugs to known sites on antibodies, none of which are especially easy to pick up and adopt straight away in your own lab. And so we've developed our own approach to site-specifically label antibodies. This takes advantage of the fact that IgG antibodies have a conserved structure. And part of this conserved structure is a glycosylation profile, shown here in green, on asparagine 297 on both of our IgG heavy chains. So we can use this conserved domain as a handle to which we attach payloads, whether that is a drug or a dye or another payload of your choice. We call this the SiteClick antibody labeling system. If we look in more detail at this branched carbohydrate domain on each heavy chain, the SiteClick antibody labeling system provides one enzyme, a specially engineered beta-galactosidase variant that we've developed to specifically remove the terminal galactose from this branched carbohydrate. And we proceed with a second enzyme, again, specially engineered beta-galactosyl transferase, which replaces our N-acetyl-galactose with a synthetic sugar containing an azide tag. And now, once this azide tag is incorporated into our antibody, this conjugate is now stable and can be set aside for later use. And when you're ready to perform the conjugation, you procure an S-dibo conjugate with your label of choice on it. S-dibo is a strained alkyne copperless click reagent. And so it will specifically recognize the azide we have introduced to our antibody. And so we now have a covalent link between our payload of choice and the heavy chain of our antibody. And so at a high level, here's an overview of this reaction where we take an IgG antibody raised in a mammalian system so that our carbohydrates are present, proceed through azide activating it, and then use an S-dibo alkyne to attach exactly 4 payloads per antibody. And so we can show that not only is our conjugation site far away from the antigen-binding domain of our antibody, which maintains the integrity of our antibody as well as its affinity for its target, but we have a reproducible and reliable number of drugs or dyes per antibody. We can quantify this using mass spectrometry, where here we conjugated monomethyl auristatin E, a toxin, to Trastuzumab. And what the mass spec shows us is that the primary species in this conjugation contained 4 of our drugs. A small subset of our antibodies had only 3, but our smaller species are almost not present. And we see this again and again. This is a reproducible conjugation where in this figure we looked at 28 different antibodies with varying isotypes, varying specificities, and species, and we see less than 10% variation across all 28 of these unique conjugations. So provides a great deal of reproducibility in a protocol that was using other chemistries is known for its variation. Now, by binding specifically to the Fc tail of our IgG, we can show again with mass spectrometry that before and after conjugation, we have seen no change to the antigen-binding domains of our antibody. So even though it contains a toxic payload, we have not changed the structure of the antigen-binding domains. So SiteClick antibody labeling consists of an antibody azido modification kit, which will get you as far as the azide modified antibody, and then at that point you can choose from a list of different S-dibo conjugates to attach to this antibody. So a few different alexafluors, of course our IFL pHrodo red S-dibo, and then a toolkit of other compounds so that you can attach really whatever you would like to have on your antibody. It's the S-dibobiotin, making it possible to attach streptavidin or streptavidin-coated magnetic beads, and the S-dibo amine and amine-reactive serving as linker reagents to conjugate whatever reactive dye you like to an azide-activated antibody. And so at a basic level, if we use alexafluor 488 or alexafluor 647 and conjugate those to our model antibodies that we discussed previously, we can see that an alexafluor 647 S-dibo conjugate of our Gazyva will specifically recognize the CD20 antigen on our Ramos cells. So in a standard cell binding assay, in this case with suspension cells analyzed with flow cytometry, we have a very specific signal with low background in all of our controls, just to show that this performs as we'd expect. We can see something similar with imaging in a high content analysis experiment. Again, a cell binding assay, just asking how well do our antibodies recognize the target cell. In this case, with Herceptin labeled with alexafluor 488 S-dibo, again we see a clear positive signal showing binding and recognition of our HER2 positive cells, while our negative controls are all minimal fluorescence as we would expect. Now, we can go beyond the alexafluors and actually combine 2 things in a conjugation reaction. So for this experiment, we used monomethyl auristatin E S-dibo, which happens to have a Herceptin cleavable linker, so that when this antibody enters a target cell, the toxin will be freed and able to be released and attack the tubulin in the target cell. Except for this conjugation, we made a cocktail of our MMAE and our Lexaflor 647. So the product is a dual labeled antibody, or essentially a fluorescent antibody-drug conjugate. So when we apply this to cells, we see in our HER2 positive cell line, our Lexaflor 647 gives us a bright signal both on the surface of the cell where it is bound to the HER2 receptor, but we also see signal throughout the cell where that antibody has internalized. While on our HER2 negative cells, our signal is really quite minimal. Now, with Lexaflor 647 being always fluorescent, we can see it both outside and inside the cell. If we just want to know whether our antibody-drug conjugate was internalized, we can switch from a Lexaflor back to our IFL-Frodo dye. In this case, we're using the IFL-Frodo red S-dibo compound for SiteClick labeling of Trastuzumab. Now, when we apply this fluorescently labeled antibody to a spheroid of SKBR3 breast cancer cells, after 72 hours, we see that this antibody has penetrated all the way through this very large spheroid and not only reports the internalization of the antibody, but we have a second probe here, a caspase sensor. We see a couple of green spots showing us spontaneous death of a couple of cells in this spheroid. Now, if we take the same antibody and instead of attaching just the IFL-Frodo red S-dibo, we now make our dual labeled conjugate, so labeled with both our MMAE in a clickable version and the IFL-Frodo red S-dibo, essentially making an antibody-drug conjugate. Apply this to the same spheroid, and now what we see is not only red signal reporting directly the internalization of our antibody into our target cells, but we also see very bright green signal from those cells that have been killed by this attached toxin that has been cleaved upon entry into a lysosome. And so again, this is a live spheroid as imaged on the CX5 high content analysis platform. So a very powerful tool, being able to conjugate one or multiple compounds to an antibody without affecting the affinity or performance of that antibody. So two different methods to covalently attach a payload to an antibody and use that to report internalization of that antibody. Both of these approaches, being covalent, require some amount of cleanup, which is useful for a small number of antibodies. But if we're screening antibody clones for those that internalize, we need a different approach. And so let me tell you about Zenon IFL-Frodo, which is designed for fast and scalable screening of antibodies to determine which clones internalize. Now, Zenon IFL-Frodo is a specially engineered Fab fragment that we have developed and pre-labeled with either our IFL-Frodo green or IFL-Frodo red dyes. Critical that the IFL-Frodo was used to make these to maintain their solubility in solution. And so in an experimental workflow, you'll take the Zenon IFL-Frodo reagent, add it directly to your antibody, and incubate for 5 minutes or less. And the high affinity Fab, which is specific for the FC of your human or mouse IgG, will very quickly associate and label your IgG. And so now, whether this is one antibody or 96 different antibodies, we can add this labeled antibody to cells in whatever scale you desire. So label your antibody, add to cells, and incubate for the required amount of time. We saw with Herceptin that was in the range of 18 to 24 hours. Some antibodies are internalized much faster. But then after the required incubation time, take your cells and with no washing or cleanup required, put your live cells into either your flow cytometry system for suspension cell analysis or into a high content imaging system to analyze internalization into adherent cells. And so what we see in the case of suspension cells analyzed with flow cytometry is if we use the Zenon IFL Frodo red to label rituximab, we only see this bright positive signal when both the antibody and the Zenon are present. If we add only the Zenon Frodo red, as you see in the middle column, we have a minimal signal, which makes sense because we are incubating our cells for 16 hours with a 50,000 molecular weight structure that can be non-specifically endocytosed. So this is an important control in this experiment to be sure that it is your antibody that is delivering the Zenon IFL Frodo compound into your cells. And so we have a very clear signal showing us internalization of this labeled antibody into our cells. Now for screening, in this case, we're using a very well-behaved IgG1, but early in a development campaign, you may not know the isotype of your antibody. And so we've designed this system to be flexible, such that the Zenon Frodo IFL reagent will bind to all subclasses of IgG. In this case, we're showing an ELISA demonstrating binding of the anti-mouse Zenon to 4 different isotypes of mouse IgG. For the human Zenon IFL Frodo, we can demonstrate the same result, very high affinity across isotypes, so that whether you know your isotype or not, whether you're coming straight out of a hybridoma or you have a purified antibody, it doesn't matter. This performs across isotypes. And so this is cell-free affinity data. Let's see how this behaves actually in cells. Now, if we switch instead to a cell-based assay and now use our Zenon IFL Frodo green to label rituximab, we see a clear fluorescent signal significantly brighter than the cells alone as a background or our Zenon alone as our control. So therapeutic grade rituximab performs as we'd expect. But what if we subclass switch this rituximab and recombinantly produce another version of IgG1, but also the 2, 3, and 4 versions of rituximab? And as we see in all cases, we report very robust internalization, except for the type 2, which we expect to be less, as we saw in our earlier model with Gazyva. But in all cases, we robustly report the internalization of those antibodies regardless of subclass. And so the models I've shown you so far are very well characterized and widely published with rituximab and Herceptin. And so you may wonder, what if how does my own antibody perform in this type of assay with this system? And so we chose an antibody out of a catalog, a standard research antibody, which is actually a mouse anti-human antibody that targets the transferrin receptor on the surface of cells. And we decided to screen this antibody using our Zenon IFL Frodo internalization screen. And what we see is as we increase the dose of our anti-transferrin receptor antibody as applied to our Ramos cells, here for only 2 hours, we found that this this specific antibody, and especially the receptor it's targeting, is very active. And so we get a strong signal in only 2 hours with this model. But we report a very bright signal to noise, where the black line here is our background of cell fluorescence alone. And as we increase from left to right the concentration of our primary antibody, we have a very broad dynamic range of that antibody, at which this assay gives us a clear signal of internalization. Now, with the Zenon IFL-Frodo reagent, it's possible to increase the amount of that xenon per well and achieve a brighter signal, as we see with the red line. Or if economics is a critical factor, you can use less xenon, as we see with the blue line, and still have a clear indication of internalization of your target antibody. And so today we've looked at 3 different methods for antibody internalization. Our IFL-Frodo red and green reagents are for amine-reactive conjugation and monitoring of internalization. The SiteClick system is designed for site-specific labeling and covalent modification of antibodies, while the Zenon IFL-Frodo is designed to screen many, many different antibody clones. And so for more information about the things we've discussed today, I'll direct you to thermofisher.com/ADC discovery for more information on the protocols and the way we generated the data in this presentation. So thank you very much for joining us today, and we appreciate your time. Thank you.
Thank you, Chris, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, can the new IFL-Frodo dyes be used to label proteins other than antibodies?
Yes, thank you, Judy. That's a good question. So, yes, the new IFL-Frodo dyes are great for labeling any globular protein, where the legacy Frodo red and green dyes are useful for labeling small molecules or particles or beads. The new IFL-Frodo dyes are optimized for labeling protein, especially suited for large proteins such as antibodies, but not specifically antibodies. It could be something like transferrin or BSA or other large globular molecules. They very much excel for these larger proteins.
Well, thank you. The 2 enzymes you mentioned for SiteClick conjugation are used for other research methods. Can I use any beta-galactosidase for this reaction?
Yeah, it's true. So the beta-galactosidase is used for many protein chemistry and biological research methods. In this case, the 2 enzymes, the beta-galactosidase and the galactosyltransferase, are specifically engineered for this purpose. So not just any enzyme can perform this function. We've actually had to do a significant amount of engineering to create versions of these enzymes that will perform in this manner. So thank you for the question.
Well, thank you. We have time for one more question. Do I need to remove carrier proteins or stabilizers from my antibody before conjugation with these techniques?
Yes, so thank you for asking. That's another good question, where with the amine-reactive IFL-Frodo red and IFL-Frodo green dyes, these will label any protein that's present in solution. And so if you have BSA or FBS or another carrier protein in with your antibody, you'll want to remove that before doing your conjugation step. With the SiteClick system, there's no need to remove BSA or other carrier proteins. There is a buffer exchange step built in to put your antibody in the correct buffer and the correct pH for labeling. But it doesn't matter if you have a carrier protein such as BSA present in your antibody for the SiteClick reaction to work. So it is very flexible in that regard. With the Zenon labeling, it's much the same, where you could have a whole suite of things present in your antibody, but because of the high affinity and specificity of the Zenon reagent for the Fc domain of IgG antibodies, there's again no need to clean up those antibodies either. They can be directly labeled in whatever solution they may be in, and proceed to your cell-based experiment. So good question, thank you.
I would like to once again thank Chris Langsdorf for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May 30th, 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.